In modern medicine, regenerative science like organ transplants and skin grafts are becoming more and more common. However, these techniques often have limitations. Organ transplants can be rejected by donors, and scientists can only create thin layers of tissue for skin grafts. But what if we could mass-produce organs with cells specifically built for you? What if we could engineer cells for transplantation that acted like your own, because they are? Let's explore the development of iPS cells in the field of epigenetics. Okay, so what are iPS cells anyway? iPSC stands for Induced Pluripotent Stem Cell, where induced means created artificially. Stem cells are specific cells in your body that have the ability to turn into or differentiate into more specialized cells. For example, a stem cell can turn into a skin cell, or a red blood cell, or a neuron. A cell's ability to specialize into different cell types is called cell potency. There are varying forms of potency, from totipotent cells, which can turn into every cell in an organism, to unipotent cell, which can only turn into very specific cell types. Like most stem cells, iPSCs are pluripotent, which means they can turn into nearly every cell in an organism. But what sets iPSCs apart from stem cells is how they're made. Unlike stem cells, which need to be extracted from the body, iPSCs are created from regular somatic cells by reversing the specialization process, making them way faster to produce. Essentially, they're artificial stem cells. The process of developing iPSCs was created by a lab under Japanese researcher Shinya Yamanaka and Kazutoshi Takahashi, who were able to reprogram mice cells into iPSCs. The research team analyzed transcription factors, looking for the specific genes that determine cell potency. The team took a list of 24 factors that were known to have some relationship with cell potency, narrowing down to 4 factors that were the most effective at creating iPSCs. Using retroviruses to change these specific genes, the lab used a process called reprogramming to change the mice cells into iPSCs, later using the same process to reprogram human cells. Reprogramming edits epigenetic marks found on DNA during cell development. You see, in addition to the regular genes on DNA, there are also small chemical tags attached to the genome. These epigenetic tags can amplify or dampen the existing genes when they are being read and transcribed into RNA and proteins. Reprogramming removes or adds epigenetic marks, such as the methyl groups attached to DNA strands. By changing these epigenetic factors or by modifying the histones that DNA strands are wrapped around on, scientists can change the way which genes are expressed without altering the actual DNA sequence. Yamanaka's team used reprogramming to erase the epigenetic tags at specific gene sites, reverting the original cell back to its earlier stem cell form, creating a pluripotent cell. iPSC technology is extremely promising, with massive potential in regenerative medicine. Their ability to replicate and differentiate into any cells makes them excellent at rebuilding tissues harmed by degenerative diseases. One day, scientists may even be able to recreate whole tissues and organs, all safer and easier to produce than regular stem cells.